Bismillahirrahmanirrahim. This is the second video on the March 23 exam. Coming to question number 23, the PDX1 protein is found in a wide range of animal species, where it is regarded as essential for normal metabolism. In sand rats, the gene coding for the PDX1 protein has a much lower proportion of A and T nucleotides than PDX1 gene from other animals. Finding the complete DNA sequence of the PDX1 gene in sand rats has been difficult. Sequencing involves splitting of nucleotides. So we have got A, we've got T on this side, we've got G on this side, C on this side. So it is difficult to split it. Finding the complete DNA PDX-B in sand has been difficult. Sequences involve splitting of nucleotides one at a time from a single stranded DNA. First you've got to do it, then you've got to cut each one of these from a single-stranded DNA. So splitting of nucleotides one at a time from a single-stranded DNA. What could account for the difficulty in finding the DNA sequence of the PDX gene in sand rats? You've got to look at only this information given to you. The PDX1 gene is present only in a small proportion of nuclei. That is not right. That could not be possible. The PDX1 gene is transcribed only in cell cell. Now, why is this wrong? From transcription, we will get the mRNA. We don't get any information about the DNA. Plus, in order to find out the DNA, we have to split the nucleotides one at a time from a single stranded DNA. So the only answer which is correct is 3. The strength of the hydrogen bonding between the two strands of the PDX1 is unusually high. That is why the answer is D. You see, you had to come out from the question, you had to figure it out. Why the answer was this? Because it said finding the complete DNA sequence is it said here finding the complete DNA has been difficult. Why? Because it involves splitting of the nucleotides one at a time and from a single stranded DNA. So the 3 was the only thing which was correct, which made sense that this is why it is difficult. Okay, then coming to question number 24. Different tissues in a plant were supplied with radioactively labeled substances to identify which tissues were actively synthesizing mRNA. Which radioactively labeled cell would be suitable for this experiment? You see, adenine is in mRNA, DNA, both mRNA and DNA. Uracil is only in RNA, and ribose is only in RNA. So that is why we had to understand why C is the answer. Because we wanted to identify the RNAs, mRNA. RNA contains uracil and mRNA contains ribosome. But you see, DNA contains adenine and also adenine is present in RNA. So the things which were only exclusive to RNA, we had to identify that. Then question number 25, which row correctly matches the structure and function of phloem sieve tube elements? Now A is peripheral cytoplasm with no nucleus. Okay, fine. To provide as little resistance to flow as possible. Yes, that's correct. A is the answer. But then why is B not the answer? B, end walls modified to form sieve plate to slow down the rate of transport of solutes. No, the sieve plates in fact allow movement very easily. It's those sieve plates which are going to allow the movement. So there is no blockage to the flow. So this was wrong. Elongated sledge joint end to end to form a tube to transport mineral ions and water. No, sorry, sieve tube does not transport mineral ions and water. Then cellulose cell wall with no lignin. Yes, that's in the xylem, we know that. To prevent loss of water, why cell wall is fully permeable? So 25, the answer is A. Question number 26. Which features of xylem vessel elements helps adhesion during transpiration? Adhesion is, of course, the ability of the water molecules to stick to the sides of the vessels. Lignin forms a complex secondary wall. No. New vessels carry extra water as the plants grows. No. There are no cross walls between vessel elements. Well, that's not adhesion. Adhesion, we're talking about adhesion. Adhesion is the ability to stick to the. So the important thing was D. the vessel elements form a narrow tube. Well, if it's a very narrow tube, then the water molecules stick to the sides, and then they sort of climb. It's like somebody climbing up the ladder on a wall. There's some ladders which are stuck to the wall, and so they can just climb up on it, just like a monkey climbing up a tree. So question number 26, the answer is D, narrow tube, so that then the water molecules can stick to the sides and it helps in the movement of the water molecule in the xylem vessels. Then coming to question number 27. Question number 27, mass flow is the bulk movement of materials from one place to another. How many of the vessels listed carry fluids by mass flow? Artery, phloem, sieve tube, vein, xylem vessel. All of them. All of them carry fluid is called bulk flow. So artery also, phloem also, xylem also, and vein also. That is why the answer is D. So these were four. It says how many of the vessels. Didn't say which ones. It says how many of the vessels. Was it one vessel? Was it two? Was it three? Or was it four? If it was only one, if it was something only arteries carried blood, then it will be only one. So again, some students would have really messed this up because they would not be able to understand it. So how many of the vessels listed carry fluids by mass flow? All four of them carry fluids by mass flow. Then coming to question number 28. Cellulose, lignin, and suberin are components of various plant cell walls. Descriptions of these cell wall components are listed. A component of the Casparian strip redirects water into the symplast pathway. A hydrophobic component of cell walls. Water interacts with this molecule in the apoplast pathway. Which row correctly matches three of the descriptions with cell wall components? Now, how did I solve this question? So you see, I remembered this, a component of the Casparian strip. Now I remembered this, it was suberin. So the answer could have only been A or D. This is how I narrowed it down. Now then I read through it again and I said lignin 3, a hydrophobic component of cell walls, or could it be 4? Water interacts with this molecule in the amphoplast pathway, and then I realized no, that's not possible. So that was 3, and then cellulose, water interacts with this molecule in the apoplast pathway through the cell wall, along the cell wall. Apoplast, A along the cell wall. This is how I have taught it to you. So question number 28, the answer was D. But you have to figure this out. I would have come up with this thing. This is the one which would have given me the clue to do this question. Question number 29. The arrows show direction of water movement across the plant root form from water in the soil to the xylem. Which arrows show water movement only in the apoplast pathway? Now, if you remember, apoplast pathway is through the cell wall. So it can only be 2 and 4 because in 6 what happens is it goes through the cell wall but then it enters the cell. So that is why the answer is C. Through the cell wall. Only, or I say along the cell wall or through the cell wall, just to make you all remember it. Then coming to question number 30. Uh, which row correctly identifies sources and sinks of sugar? Storage cells of seed that is starting to grow has to be source. So these were the only two possible answers. Root cells absorbing mineral ions have to be sink. So that is why the answer is A. Can't be. Both are not source. Well, of course they can be. I'm not saying that can't be. They can make a question like that and both can be the source. But here it says which row correctly identifies source and sink. Root cells absorbing mineral ions is a sink. It needs the glucose from the source. Question number 31. The photomicrograph shows a section through a tissue with an artery. Now you can see around these are alveoli. This is lung tissue. Which row correctly shows the type of artery and whether the blood inside the artery is oxygenated or deoxygenated? In the lungs, we have the pulmonary artery. So the pulmonary artery brings deoxygenated blood from the heart. So the pulmonary artery in the lungs, deoxygenated blood type of the artery is in the organ, so it has to be muscular artery, and it has to be deoxygenated blood, so it has to be A. Muscular, why? Because I told you we've discussed this in many previous videos, is muscular arteries are those which are nearer the organ, because then the muscle is going to contract or relax and will increase the pressure to that organ, the blood flowing with greater pressure into the organ, because as the aorta leaves, the aorta has less muscle, but as the arteries go near the organs, they have more muscles in them. So muscular artery, and the fact that it's in the lungs, and artery in the lung is the pulmonary artery which carries deoxygenated blood. Question number 32. Heart surgery may cause a decrease in the transmission of impulses in the Purkinje tissue to the right side of the heart. Right side of the heart. What is a possible effect of this decrease? Impulses would be delayed by the AV node. The muscle of the right ventricle would contract slightly more slowly than the muscle of the left ventricle. Yes, that's the answer. Because the Purkinje tissue supplies the wall of the ventricles, and the ventricles then contract in response to the electrical impulse in the Purkinje tissue. Now, why is this wrong? The muscle of the right atrium would not contract because the Purkinje tissue doesn't go into the atrium, and the sinoatrial node would transport field that is again in the atrium. The SA node is in the atrium. It's nothing to do with the Purkinje tissue in the right side of the heart. I'll just show you a diagram to see if you remember what is the Purkinje tissue. You can see here Purkinje tissues where this is the Purkinje fibers. Another word for it is also the Purkinje fibers. So these are the Purkinje fibers, and these are the Purkinje fibers. And these are what we need to understand. These are the Purkinje fibers. So they supply the wall of the ventricle, and they are the electrical, the conducting system as we call it. So these are called Purkinje fibers, or Purkinje tissue is also the word used for it. Now coming to question number 33, the diagram shows a network of blood vessels that supply blood to muscle tissue in the human body. So blood vessels to the muscles in your legs or muscles in your arms. What is a correct comparison between the blood at X? So this is an arteriole, and blood at Y, that is would be the capillary network when the muscle tissue is at rest. So blood pressure would be higher, definitely. That is, so it has to be either this or this. Now the question is, would the water potential of blood at X be higher or lower? Well, it has to be higher. Why is it got to be higher? Because the arteriole would have more water, but when it reaches the capillary network, then water and glucose and everything is going to move out. So the water is also going to move out. So the water potential at X will be higher. Here it will be higher than Y, because it says water potential is as compared with water potential at Y. So this is what we have to understand, and that is why the answer is C for question number 33. Now coming to question number 34, scientists have shown that the oxygen dissociation curve for hemoglobin of smaller mammals are to the right of the larger mammals. So this is the oxygen dissociation curve, and it says that the smaller mammals is to the right. So this means it is towards the right. Now this is of course the Bohr shift. This is the Bohr shift when the reason is that you see too much carbon dioxide, so that means the hemoglobin will give up its oxygen, so the percentage saturation will decrease. So of course that is why the answer to 34 is C. It releases oxygen more easily than the hemoglobin of larger mammals. So they've converted the Bohr shift story into another story, and they're checking whether you can figure this out or not. I mean, if you are clever enough to figure it out or not. So the rest are all wrong. Then coming to question number 35. What is an effect of an increased concentration of carbon dioxide in the blood? Now coming to question number 35. What is an effect of an increased concentration of carbon dioxide in the blood? Now if you remember, carbon dioxide increases, then it combines with water to form carbonic acid. This dissociates into hydrogen ions and bicarbonate ions. Hydrogen ions combines with hemoglobin to form hemoglobinic acid HHB. Hemoglobinic acid. Sorry, I shouldn't be using this HGH. Hemoglobinic acid. So this would be HHB hemoglobinic acid. So that is why the answer is B, increased concentration of hemoglobinic acid. Why the other wrongs? Increased movement of chloride. No, bicarbonate ions move out of the red blood cells, chloride ions move into the red cells. Here it is saying out of the red blood cells, so this is wrong. Then decreased concentration of hydrogen carbonate in the blood plasma. No, they'll be increased because the bicarbonate ions will move out and chloride will move in. Decreased concentration of carb amino hemoglobin. Why? 5% of it is always carried as carb amino, 5% is CO2 in the plasma, and 10% is carb amino hemoglobin. And then the 85% is the bicarbonate ions. Question number 36. Two of the requirements of an efficient gas exchange system are a large surface area and a short diffusion distance. Which row correctly describes how alveoli are adapted to meet these requirements? Well, of course, first let's look at the answer and then look at the ones why the others are wrong. So the answer for 36 is C. Alveoli are folded and interconnected, and walls of the alveoli are only one cell thick. Now, large surface areas achieved by the alveoli by millions of alveoli. Elastin fibers prevent the alveoli wall from collapsing. Why is this wrong? It doesn't create a large surface area. On short diffusion distance, an extracellular layer inside the alveolus wall contains blood capillaries. Now, this doesn't decrease the diffusion distance. Gases dissolve in a layer of liquid to speed up diffusion. This does not increase the surface area. Then, walls of the alveoli are formed of squamous epithelial cells. This means the wall of the alveoli is very thin, but that doesn't make it in any way increases the surface area in any way. So that is why these are wrong and the C is correct. Then coming to question number 37, what defines infectious diseases? Now, what is an infectious disease? First, let's get this right. Infectious disease in which a pathogen is transferred from one person to another. And there are many infectious diseases. We can't just talk about one. So symptoms are caused by a bacteria that passes from contaminated air, soil, or water to a host. Symptoms are caused by a pathogen that is transmitted from one host to another. Yes, B is the answer, but why are the other wrong? Infectious diseases are not all caused by bacterium. Infectious diseases, there are also fungal infectious diseases, there are also viral infectious diseases. Symptoms are caused by a microorganism that is carried by a vector, not necessarily. All diseases do not need vectors. Symptoms are caused by a virus that mutates to infect a new species. No, why would that be? I mean, infectious diseases can be of any microorganism. So basically, we say the definition is caused by a pathogen. We don't say bacteria because even a bacteria is a pathogen, even a virus is a pathogen. Even some fungi are also pathogens. So the answer was B. Now coming to question number 38. Question number 38. A hurricane destroys a large town on an island. People move away from the town and set up tents where sanitation is poor. Sanitation means sewage disposal and all that is poor. There are no toilets. People are just defecating and urinating anywhere and everywhere. Flies sitting on it, flies sitting on the food. So, which disease is most likely to spread within a week of the change in living conditions? Of course, it's cholera. That's the only thing which was going to spread. It's waterborne and foodborne. So, 38 is A. Then coming to 39. Which statement correctly explains why viruses are unaffected by penicillin? Now, why viruses are unaffected by penicillin is penicillin only affects host cell metabolism. Penicillin only binds with 70S ribosome. Virus does not have any 70S ribosome. Please remember that. Penicillin only blocks mRNA synthesis in prokaryotes. But it didn't say that. It didn't say what is the role of penicillin. It says which statement correctly explains why viruses are unaffected by penicillin. Why? Because penicillin only blocks the peptidoglycan synthesis. And in viruses, there are no peptidoglycan which need to be synthesized. It has a protein coat and there's no cytoplasm. And then it has an RNA or a DNA strand. Now, coming to the last question, question number 40, which process characterizes the mode of action of phagocytes? The first thing which is wrong is antibody production. Lymphocytes make antibodies, so anything with one in it is wrong. So this one is wrong and this one is wrong. Now we're left with these two choices. Now, do phagocytes have a receptor binding, endocytosis, exocytosis, and hydrolysis? Yes, it's right. So that is why the answer is C, which is why? Because they bind, they have receptors, they bind to it, and then they will cause endocytosis, and then, of course, exocytosis, and then the hydrolysis and all that. So 2, 3, 4, and 5, and the answer is C. That completes this latest paper, and I hope this is helpful to you all. But please remember, MCQs are very tough, and you need to score very well in the MCQs because we have not done any paper 2 and 5. Sadly, they were cancelled because of the situation that we had in our country. So please be very careful with the MCQ paper and try and do it very diligently and very carefully.